Hello everyone, I am Rakshanda Sajeed and I'm representing my team from TCS Research India, and I'm honored here today for this opportunity to present our work on an improved deep learning model for immunogenic B epitope prediction. So, a brief introduction to the world of antibodies and antigens. I think we all know it, but still, I would like to introduce that way. So, antigens are foreign proteins or fragments which enter the host body via infection and are capable of eliciting immune responses. And a specific small site on the antigen to which a complementary antibody may specifically bind is called the epitope. And these epitopes are usually 5 to 8 amino acid residues on the surface of the antigen and can elicit a highly specific immune response. So, the prediction of these epitopes is very crucial for the development of therapeutics and vaccine subunits. And the development of computational methods for the prediction of these epitopes has thus been going on for quite some time. In this end, the language models have proven to be very, very promising. And these are a few tools which are popularly used. Out of these, BepiPred3 and Discotope3 are very popular and state of the art. BepiPred3 uses embeddings, protein embeddings from ESM2 for the prediction of B epitopes. However, we think further improvements in precision are necessary for practical applications. And we also think that these protein language models might not be able to capture certain structural features which are important and which involve protrusion and accessibility in the proteins and are important for the prediction of B epitopes with more precision. Thus, our work hence involves utilizing the embeddings from ESM2 and combining them with certain structural features of the proteins for the prediction of these B epitopes. And we've also explored different ways of concatenating these structural features with the ESM embeddings. And we observed that the method of deep concatenation of these features works better. And I'll be talking about it in details in my further slides. First, now coming to the data set that we have used. So for our work, we have used the B-cell receptor assay data from IDB 3D for training. And we have used a benchmark data set used by Serendip CE tool as our independent test set. We filtered these antigen sequences based on their length and also the resolution of the complex structure. We've also made sure that the training and the independent test sets are no more than 25% similar to remove redundancy. And the structural features that we have used for our work are contact number, protrusion index, and half sphere exposure. And these features represent the locality or the spatial information within the protein, especially in the interacting area between the antigen and the antibody. It's the shape of the antigen and also the accessibility of the amino acids around the interface. Thus, we have proposed two different kinds of neural network architecture-based models for this classification tasks. And in both of them, we have used ESM2 as the feature extractor, which gives 1280 dimensional feature vector for each epitope or non-epitope data point in our data set. So this here is the first architecture. Where the 1280 features are simply concatenated with the structural features, different combinations of the structural features. And this way of mixing these different feature sets is commonly seen to work well in all settings. And here the final output representation passes through a 4-layer neural network. And finally, where the weights are learned and for the classification of a residue to be an epitope or a non-epitope. Then coming to the second architecture, here the ESM2 embeddings are first transformed through a 4-layer neural network. Then the transformed feature vector is concatenated with different combinations of the structural features. And this combination finally passes through the final layer of the neural network to give whether the data point is an epitope or a non-epitope. So working with these two architectures, we compared our models to different state-of-the-art tools available. Here we have shown 4 tools: BEPI-PRED2, 3, Discodope 2 and Discodope 3, and the comparison with our models. For each tool, we calculated the prediction values for the entire protein sequence on each amino acid on the independent test set. And we compared the models using the ROC and the PR curves and scores. And we can see that the deep concatenation of features, that is the architecture 2, fares better than the naive way of concatenating the ESM features with the structural features. And also, we try, since there are certain models with which have structure aware which are structure aware, like SAPROT. So we compared our models even by taking embeddings from SAPROT and concatenating them with the structural features that we have taken for our work, and we saw that still ESM2 embeddings concatenated with the structural features, it fares better as compared to the SAPROT embeddings. And this maybe we can infer from here that these features are actually quite significant in this classification of the B epitopes and its prediction. So okay. Here, this is the table where we've highlighted the different metrics on which we've compared the different models of both the architectures as well as the different tools that we have taken into consideration. Here also we can see that the deep concatenation method still surpasses the ensemble way of using the features. Now coming to an interesting analysis that we did for our work. So studies show that CD4 T helper cells aid in selecting high affinity B cell receptors, and this is a process termed as TB reciprocity. And we know that ESM2 captures the physicochemical properties of proteins, the evolutionary contacts, and also the conservation of amino acids. And there have been studies where they have included TB reciprocity as a feature to improve the B epitope prediction. And this got us thinking if, with the help of an attention analysis, we can assess whether TB epitope proximity is captured by vanilla ESM weights. The idea being that with the plethora of protein information provided as context to ESM, the pairwise residue information for T and B epitope residues captured from the attention matrix should be statistically significant. So for this analysis, what we did was we shortlisted 156 antigen sequences from IEDB, which had both T and B epitope annotations. We extracted the attention matrices for these antigens from the last layer of ESM2, and by averaging them out across all its 20 attention heads. And each element of the attention matrix of an antigen represents the degree of the can I, yeah. Yes. Is it? Yeah, it's okay. Degree of importance that a residue gives to the other residues of the same antigen. So here in this expanded figure, so this is the sequence. Here in the y-axis, we can see the T cell epitope residue, for example, E, and which are the residues, B cell epitope residues that it is giving higher attention to. Here on the x-axis, we have highlighted. So that way from each, so taking this example, for E, we have taken the vector from the attention matrix for E. We've sorted it in the descending manner, high to low, giving high priorities, and that way we taken all the residues, B cell epitope residues, which are highly attended to by this particular epitope residue E. And to check whether this result or these results that we've gotten, whether they are just random or they are statistically significant, we considered two extreme cases. We considered those sequences where none of the B cell epitope, known B cell epitope residues were attended to by the T epitopes, and such cases where all the B epitopes were attended to. And we did the same analysis even for linear epitope data. We did it for conformational epitope data as well as linear. So in both the cases, we can see that there was significant difference between the representation of prediction scores. These prediction scores are ESM scores itself, and there was significant difference seen. And yeah, so the plot actually highlights that. So we were a little optimistic, and maybe this is a very small set, I agree, but maybe with further analysis, this is something which can be worked on. We found it quite interesting. So to conclude, our work shows the protein embeddings that combined with structural features, and it leads to improved prediction accuracy as compared to protein embeddings alone. And also that the deep concatenation architecture performs better than the naive way of concatenating different structural features and the embeddings. So also highlighting the importance of these features, contact number, protrusion index, and half sphere exposure. And also our study shows the TB reciprocity captured by ESM2 to an extent, and also it might help in prediction of high scoring immunogenic epitopes. It can be studied upon. Well, thank you.
Thank you very much. Do you have questions from the audience?
One of the classic problems of B-cell epitope prediction cast as a binary classification is how to define the negative data. So like for conformational epitopes, I mean, the positive data are usually like the contact residues in a monoclonal paratope complex with its epitope, right? But how can you define like negative data and be sure that it's not just a subdominant epitope that's being masked in the presence of a more immunodominant epitope?
Right. For our work, as you said, the epitopes are very specifically defined in IEDB, right, from the data set. But for our work, for the negative data set, all the other epitopes, all the other residues in the antigen sequences, they were taken as negative, and from there we just randomly picked out so that we balance the positive number of residues with the negative.
One potential problem there is like you might be defining sets of residues that aren't really plausible as paratopes. So it might not be a fair comparison because the randomization might generate something that's like unphysical in the sense that a real paratope might not be able to span those residues or like bind or contact them all at the same time. So that's something that you might want to.
Yes, of course. This is one problem. I think most of the studies do this, taking the like the way we did. But yeah, this is definitely one bias that brings in the analysis. That's true. Agree.
Next question.
Thank you for your talk. So my question is it possible your model will generate multiple potential epitope for same antigen? Like if yes, which one is right? Or maybe they all correct? Since COVID antibodies can binding into different epitope for the same antigen. So how do you handle this situation?
If I got you right, so are you talking about antibody-specific epitope prediction?
Yeah, kind of like same antigens, they may have multiple epitope, right? Like if your model generates the epitope for this same antigens, like maybe there are potential like two different epitope, like when you evaluate your model or something, how can you define which is the right? Maybe they are both right. If you only choose one, are you going to missing the other one?
At the moment, our model does not do antibody specific. For that, we can actually do it by giving antibody information, feeding antibody information, and then maybe modeling it that way. So maybe then we can get, give importance or underscore certain epitopes which are cognate to that particular antibody, I think.
We have an online question, I guess.
Yes, Dor Goslin wants to know what is the size of each protein, epitope, encoded, how many amino acids?
So these are conformational epitope rests. So we have taken it residue-wise and not as an epitope as a whole. So it will be around 5 to 8 regions on the antigen, if that is what it is. Yeah.
More questions online?
No, that's it.
Questions here? In that case, we thank the speaker.